There's no room for hate.
Where are we going? In the store?
Can I understand why people would? Yes. I'm starting to live a life.
Welcome to the Crick.
Oh wow. Yeah.
And I mean, it is quite a place, isn't it?
It is, yes.
Kathleen's story is important, and it's important to tell the story because it shows that science can go further than, routine diagnostic investigations.
It's amazing.
So isn't it incredible?
It's vastly.
It's just, it's kind of surreal
Like
for
all
me.
that. It's like how...
When you're dealing with deaths or difficult diseases that are hard to diagnose or to understand, science, and in particular genetics, can illuminate answers.
It's a good thing to be able to stand up and say, science did this, science created an answer, science gave me my peace of mind, and science helped me sit here today to be able to talk to everyone else.
That's why we're here tonight, because we really want to make a difference. We want to build that intellectual capital, and because it is hard. And did I ever intend to be an expert in this stuff? No, not at all.
Did I intend to be in prison for 20 years? No.
No.
I did not.
No.
I didn't know Kathleen. I was hopeful that, I thought, maybe you can find something. I was just curious, I guess. I studied a combined degree in Australia at the Australian National University of Science and Law. I did my science honours in immunogenomics. For as long as I've worked as a lawyer, I've wanted to try and stay connected to that science element. Had no connection to the case other than just watching a news story and going, I think this would probably be a really good candidate for whole genome sequencing. Genetic sequencing had progressed a lot since the point of Kathleen's conviction in 2003. Cost had dropped a lot. Whole genome sequences was really taking off at that time. So I thought, why not try and see if you could sequence the children and find out if there was a genetic cause of their deaths. So I Googled the lawyers that were acting for her, and I called them up and I said, have you thought about whole genome sequencing? And they say, that sounds interesting. Can you organise us an expert to do it? And I said, well, I can look around. I got turned back from a few, but then I reached out to Professor Carola Vinuesa. I don't think I realised how lucky I got at that time.
I remember Dave calling me and telling me this story that I felt a little bit embarrassed that I didn't know about this case. I remember you telling me that at least 2 of the children had some clinical disease. One of the children, he told me, had epilepsy and had died presumably during an epileptic fit. And I remember the case of the last child to die, and he told me she had been found to have myocarditis on autopsy. I was surprised that children with these kind of diseases, it could have been concluded that these were murders. Dave was suggesting, can these children have whole genome sequencing? And I remember saying, well, it can be done. I think you also were querying whether...
Well, we didn't know if there'd be samples. I mean, Caleb died in, 1989?
Yeah, it was 30 years before, right?
Yeah.
So you were a bit worried about that and about whether the quality would be good enough, right?
I guess finding you was sort of like, striking gold in terms of finding someone that was interested and also not just interested from the science side, but like the practical side of things. We were essentially trying to perform a genomic autopsy and trying to find what is the administrative pathway to doing that.
I said, well, look, if nobody wants to do it, we could do it from a research point of view. I thought it was preferable to do this as part of a team so as to not overlook anything.
The friendship started back in public school, in Katara Public School. I was already there, and then Tracy came along. We've been inseparable pretty much ever since. When tragedy hit me and I ended up inside, she was the first and only one really that stepped up and said, okay, this doesn't seem right. You've got my friend in prison and this is wrong.
I thought of contacting one of my colleagues, Todor Arsov, because first, he's a very exceptional mind. I knew he also cared about justice, and he would go the extra mile when there are problems. And this didn't seem a very straightforward case.
The biggest problem in genetics is getting to test somebody which has signs or symptoms of a condition. Just knowing the story that these are children that have died 20 years ago, it was really problematic that how are we going to get to DNA samples, because people get the idea that this was all kind of provided in a way, but it was all an uphill battle, nothing was easy like that. Given, Kathleen was, the only person that we could potentially have access to, we then strategized around how would we go about that and went to visit her in prison, obviously to understand what happened to the children, but to also uncover her personal history, which then provided the path forward for doing the genetic testing in that family.
And it can't be underestimated how important that was, right? Yeah. Because that gave us, you really extracted some useful information, including her history of fainting, syncopes, which in our minds meant that there was a possibility that she also had a cardiac arrhythmia, because syncopes is a symptom of cardiac arrhythmia.
It was the first time in my life I've ever been around courts or anything judicial, prisons and stuff like that, so it was a major, anxiety, I suppose. And also, because we, were having, I suppose, considerations around taking, say, blood samples, for example, but that was not possible because we couldn't take the needles through the metal detectors, and then the sick bay in the prison would not provide the service, so it was all a little bit complicated and convoluted like that. So which prompted then another visit a couple of weeks later when we could get around all of these dramas by getting the saliva sample and the buccal swabs from Kathleen, which didn't require any metal needles. And so that's how it all unfolded.
Once I had a mouth swab done when someone was actually serious and came to the prison and said, we want to take some saliva off you. And I'm like, oh, happily, here you're at. And for it to develop from there, that was quite a eureka moment, sort of thing, because that gave me the glimmer of hope that someone was trying to find an answer of some sort.
Every cell in the body contains DNA. The genes in our DNA are code for making proteins, which are vital to how our cells function. Variants in the DNA can change the code, so it now produces a faulty protein that can cause fatal diseases. We extracted Kathleen's DNA from a saliva sample and sequenced it. Out of the 20,000 genes in our genome, we looked for variants in around 350, potentially linked to unexpected deaths of children.
There were over 40 genes already that were well established to cause sudden death. But we decided to be quite comprehensive, so then not just cardiac arrhythmia genes, but as I say, epilepsy, inflammation, immune deficiencies, metabolism, mitochondrial defects. And each of us had a list. This was a Sunday. I had just come back from picking my daughters from football. We met in my kitchen, right? I remember we were on my kitchen bench.
Carola was on the right, and we were both with our laptops doing the lists and obviously coming with, candidates, favorite candidates, and I would say, oh, such and such gene, and Carola would go, yeah, I looked at that, but, this and that, and then she would go, oh, what about this one? And I went, oh, yeah, I looked at that one, but, something is amiss. And then I just remember this moment, like we're turning both at each other, like, calm too. So it was just, one of these cosmic moments for me, I remember that very, very special moment.
We found a variant in a gene called CAM2 that had never been seen before.
The one that really attracted our attention was this variant in CAM2. Our pipeline would automatically predict the probability that the variant changes protein function. It was already predicted to be probably damaging.
CAM2 is one of 3 genes that each encode the same protein called calmodulin. This crucial protein is involved in regulating heartbeat.
So we started just frantically going over the literature, looking at whether the variant was really novel, a couple of confirmations, was it predicted to be pathogenic? And I think within the next couple of hours, we were both semi-convinced that if there was something, this particular variant had to be it, because it just fitted many of the expectations we had about what we were looking for.
Previous research had shown variants in the 3 CAM genes are very rare and potentially deadly, causing specific mutations in the calmodulin protein. Dozens of variants had been documented in dozens of individual cases, many linked to heart problems and sudden unexpected deaths in children.
We all agreed that the right course of action would be to sequence the children, ideally the father as well, and we had a discussion about how to go about looking for samples.
This is all on the edge of what's technologically possible. Luckily, it was possible to find the cards in the central registry in Sydney, but then also the Victorian
Yes, the VCGS, the Victorian Clinical Genomics Services.
They were happy to take that on.
They told us that they could do it, but they couldn't guarantee that if the samples were too old, and they thought 30 years old was very old, that they could extract good quality DNA. But they actually managed to eventually, right?
Impressive.
So that was a little help from heaven, I think. You know,
Yes.
one of those instances
Yes.
where, you know,
Yes.
something that's almost impossible
Yes.
worked out.
When we sequenced samples from Kathleen's 2 daughters, we found they also had the novel variant in the CAM2 gene, causing a specific mutation in calmodulin. Then we learned of a case of a young child who had suddenly died and his sister who had suffered a cardiac arrest, both of whom had a mutation in the same position in calmodulin. This was key evidence the novel variant was likely to be pathogenic.
That should have settled it, right? You have the leader, Peter Schwartz not only found this family, he was the one that had put together the International Registry of Calmodulinopathy. So he was, as he said himself, the man with the numbers, right? He knew about the 70 cases in the world. The phenotypes were not necessarily fitting perfectly with other cases that had been reported, but he said, we've done this for long QT syndrome.
You've got 74 cases in the registry at first. Nearly everyone has some differences to another one.
Exactly, the phenotypic spectrum is expanding continuously. We are seeing different presentations as we go. In my view, this fits with what we know of how cardiac arrhythmias evolve over time. But despite his letter, which was pretty firm, right, and gave these 7 points as to why he thought this had to be looked at very seriously, it wasn't taken seriously. I remember Dave calling me and saying, the report is out, you are probably not going to believe it and like this. And I just felt, I couldn't believe it. I actually couldn't bring myself to read it because I was so convinced it was going to be a different finding. So I asked Dave to summarize it for me because I really couldn't bring myself to go through that, and I was very, very, very disappointed.
Yeah.
So much so that I remember there and then we had like 2 or 3 conversations in the day and said, what can we do to have this evidence, right, that it has become such strong evidence and that it's not taken seriously in a courtroom, right? They can't understand the significance of these last final findings.
An addendum had been written to the report dealing with that evidence, basically saying, we acknowledge the new evidence, but we don't need to reopen the hearings to hear about this evidence more. We don't need to essentially talk to the experts. And I found that quite surprising.
We had the first inquiry and that all got dashed. I said, so it was like, oh wow, we just went through all that and now what? I was just stubborn and decided stuff yous. So, when you haven't done anything, it makes no sense to me why you would roll over and just accept your situation.
What happened over those 5 years really illustrates the power of science at solving a problem, at not stopping at the first obstacle. Science can go and take that extra step and prove it. We thought that one of the ways to really prove definitively that this mutation changed the protein function and therefore could cause an arrhythmia was to do functional experiments in the lab. And for that, we asked the experts who in the world can do these assays, and they recommended Michael Toft Overgaard.
It was clear for me this was a very special case. I never worked on something that had been in a courtroom or murder case.
My background is in human genetics, and I've worked my whole life linking variants we have in our genome with disease.
Mette had discovered the first mutations in calmodulin in a family with a cardiac arrhythmia.
Since we are married and we live on the exact opposite, end of the world in Denmark, so we were kind of working alone on the reports, and it was important for us. We decided we don't take a stand on guilt, but we take this fresh look. What do we think about the variant?
Hello, how are you? Nice to see you. Welcome. How are you?
Hey, you made it! Hey, thank you.
Quite quickly decided not to look very much at the history of the case. So I think I never actually realized the whole process of the sentencing and stuff. So, but just let's have a look at this variant.
Gorgeous,
You could say in the beginning it was mostly you, Michael, who was involved in the case, and what really motivated me also to step in was how underappreciated these studies and these modern genetic concepts actually was. A lot of people were asking, but if it is pathogenic, how can Kathleen then be alive? It has to do with survivorship bias. So survivorship bias is really when you look at a group of people, or just one people who are kind of either winning or surviving, and then you mistakenly think that you are looking at everybody who took that test or carried that variant. And then you say, oh, that person survived, then everybody must have survived. And you really fail to think about that there may have been people, going through the test or carrying that variant that actually died. Mm-hmm. So it's kind of the wrong conclusion. It's a completely common concept in genetics that you would have some people, for some reason or another, could be the other genetic variants that they have, the modifier genes, could be something in the environment that determines if that person actually get the phenotype or not.
Michael, how did you approach the functional test?
We order the gene online with the variant or mutation in it, and then we take the gene and we put it into bacteria, and then we have the bacteria make the protein.
To show the novel variant's effect, we compared the variant protein with regular calmodulin in the lab. Tests showed the variant damaged the calmodulin's ability to fulfill vital roles regulating heartbeat.
We were really surprised by reading the hesitation to say this could be a dangerous variant.
I remember you read all the reports, like what are they saying, taking in a way an open mind to all the arguments. And this is basically when you said, I think this is the kind of the key point of disagreement, is that the phenotype doesn't match what was already seen.
Yeah, and then they mentioned it's only the sodium channel where mutations leads to cardiac arrest while you sleep. And when I read this, I say, well, this is not really weird, because calmodulin is also a really critical regulator of the sodium channel. I had my postdoc do the assay, and like 2 days later we had the first results.
You went into the living room and you were there for like 20 minutes and then you were like, hey, can you come and take a look? And I remember when I entered the living room, you're kind of turning your laptop and it was so clear that this variant was certainly in an important domain. So I call this the sofa moment.
When you made that final discovery, it then all fitted, right? These girls died in their sleep, but had intercurrent infections and Brugada syndrome, the sodium channelopathy, right? Typically, or is one of the few that is really triggered by a fever. So it's like seeing the whole picture.
So I remember when we all read your report, it was kind of inspired. You're like, oh, this answers the discrepancy.
There it is. Yeah. Everything you always wanted to know.
Yes.
Yeah, and I have to say that we notified the inquiry. This was like 4 days before the inquiry started.
Yes.
So we notified them and had an online meeting and showed the data, and they could see, okay, this is important.
It was the fastest release known in New South Wales court systems, and it was literally 56 minutes, which is the fastest record ever of anyone being released from a prison. So I did literally turn up in a van.
With your sand shoes?
With my sand shoes in my hand, and borrowed clothes on, none of my possessions, and it went from there. So I was living with Tracy.
Oh my God, look at you! Hey! Oh my God, look at you too! Presley, I'm so happy to see you again.
Sorry, so nice to see you.
No, it's good to see you.
I'd like to think the legal system could actually listen to scientists a little bit more often.
It's very special to have you here. I just can't believe it, Kathleen, everything that has happened, right?
Instead of accusing a woman of killing children, the first stop is, okay, oh my gosh, let's go check the scientific sides first before we go down that avenue.
I'm hopeful that a case like this can serve as a template of other cases. I think we've learned whole genome sequencing can be extremely valuable in cases where parents have been accused of killing or harming their children.
I used to wear a t-shirt all the time, especially with Carola, and that some of the publicity shots was, what do we want? Evidence-based science. When do we want it? After peer review. I just wear that at media interviews just to piss people off.
That's it, that's good, that's great.
The other thing that we've learned is that the analysis needs to involve scientists that can look at a genome, casting a very wide net. If we limit ourselves to only known variants, it's likely that we might miss opportunities of solving these mysteries.
It can happen to anybody. You can lose children for umpteen million thousands of reasons. I said, but it doesn't automatically mean that a person was involved. It happened to me. Could happen to you. Could happen to anybody. I said, so we would both hope that when it does, there's better systems in place where those people can get a bit more help than we both had. Try.